Alright, so nandito ulit tayo sa panibago nating topic all about the basics of molecular genetics. No? So in this lesson, we ask ourselves if it is related to the inheritance that we discussed in our biology class when you are at grade 8 and grade 9. We already know that the DNA contains the genes that were inherited or passed on from one generation to another, and it will be determined a particular trait. DNA molecules can be decoded to produce specific proteins which will later can be visible as a trait. No? So, maaari na natin simulan. No? So, simulan muna natin sa pinakamalaki, no? Which is yung chromosome. No? Chromosome, no, is in the nucleus of each cell. The DNA molecule is packaged into thread-like structures called chromosome. Each chromosome is made up of DNA tightly coiled many times around proteins called histones. Yung histones is isa po siyang klase ng protein that supports its structure. So, tandaan natin, chromosomes are not visible in the cell's nucleus, not even under a microscope. When the cell is not dividing, however, the DNA that makes up chromosomes becomes more tightly packed during cell division and is then visible under a microscope. Most of what researchers know about chromosome was learned by observing chromosomes during cell division. No? And then, each chromosome has a constriction point called the centromere, which divides the chromosome into two sections or arms. The short arm of the chromosome is labeled the P arm. The long arm of the chromosome is labeled the Q arm. That because the location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes. So, ayan po yung structure ng chromosome. No? So, in yung structure niya. No? So, yung maliit, tama ba? Maliit na arm. No? Ito po yung P arm. Malaki is yung Q arm. Yung nasa gitna is yung centromeres. No? From biggest to smallest. No? For chromosome, pag pinasok natin yung chromosome, meron tayong makikitang section, double helical structure sa loob niyan, which is yung DNA or Deoxyribonucleic acid. And then sa sections of strand ng DNA, mapapansin natin na meron dyan genes. No? And then sa genes na yan, meron doon nakapaloob sa kanila na nucleotides. No? At ito ay ma-encounter natin later on. No? So, in terms of history ng DNA, noong 1953, si Watson at saka si Crick ay nanalo or naparangalan, no, sa Nobel Prize Award for Physiology for their work on the structure of the DNA. Many scientists consider that the discovery of the DNA structure as one of the most significant biological events of this century. No? Hanggang ngayon, yun nga. And then the DNA structure was introduced in the last level, and this gave a clearer idea of what a gene is in terms of its molecular structure. Ayan. So, either malalaman natin yung functions ng DNA, determine a gene for a trait, and then DNA codes for a specific proteins. Let us review first the structure of the DNA, then the machinery of protein synthesis, which includes the RNA or ribonucleic acid. Ribonucleic acid, yan po yung single strand na nucleic acid. Malalaman natin siya later on. So, meron tayong mga types pa na or mga machinery pa ng protein synthesis na under ng RNA, which are the mRNA, tRNA, and then rRNA. And the site of the protein synthesis, which is the ribosome. Malalaman natin later on. And then sa DNA or deoxyribonucleic acid is a type of nucleic acid made up of nucleotides. And then it composed of deoxyribose, which contains of pentose sugar or 5-carbon sugar, then phosphate molecule, and then 4 nitrogenous bases. They are the adenine, starts with the letter A, thymine starts with the letter T, guanine starts from the letter G, and then cytosine starts from the letter C. And then ganyan yung itsura ng DNA, no? O yung internal structure niya. According to its, may tawag dito yung condensed structural formula ba yun? Correct me if I am wrong, ha? Or yung kanyang molecular formula, I think. No? Correct me if I am wrong. So, yun yung structure ng DNA. And then sa RNA naman, or ribonucleic acid, is a type siya ng nucleic acid obviously, and then it is also made up of nucleotides. Ang pinagkaiba ng RNA from DNA, it lies on the type of sugar and a certain nitrogenous bases sa nucleotide. Ito ay gawa sa pentose sugar or 5-carbon sugar na ang tawag sa kanya ngayon is yung ribose, and then phosphate sugar, and then yung 4 nitrogenous bases which are the adenine, uracil starts from the letter U, guanine, and then cytosine. Sa RNA, wala ditong thymine dito sa nitrogenous bases. Instead of thymine, pinalitan natin ng uracil. Uracil ang pinalit dito sa RNA instead of thymine. Then, yan yung structure ng RNA. And then, take note that the nitrogenous base thymine from DNA is replaced by uracil sa RNA. And then, meron tayong different types ng RNA, namely, messenger RNA, transfer RNA, and then ribosomal RNA. Yan yung ano nila, yung acronym para, or palatandaan para ma-determine natin yung types ng RNA. And then, in terms of types ng RNA, yan, the mRNA or messenger RNA acts as a sort of a message from the transfer ribonucleic acid to interpret. On the other hand, the mRNA is found inside the ribosomes. It binds the amino acids into chains which will then come out later as protein chains. The process of producing proteins with the help of the 4 RNAs is called the protein synthesis. Ito yung differences ng DNA at RNA. Yung number of strands sa DNA is 2 strands, and then sa RNA naman is 1 strand only. And then sa appearance of the molecule, and sa DNA there are double helix, and then sa ribonucleic acid, maraming mga appearances ito, just like yung mRNA, ito yung straight strand. Yung tRNA or transferred ribonucleic acid is just like a clover leaf structure, and then yung ribosomal RNA, it is packaged with a proteins to form the ribosomes, which is made up of 2 circular subunits, a smaller subunit and a large circular subunits. In terms of pentose sugar, and sa DNA, at ang tawag sa kanya is deoxyribose sugar, and then sa RNA naman, ang tawag sa kanya is yung ribose sugar. Sa nitrogenous bases, meron tayong dalawang pairs, no? Pyrimidines, no? Sa pyrimidines, sa DNA, cytosine and thymine. Sa ribonucleic acid, cytosine and uracil. Diba nga po kapag RNA, uracil, kapag DNA, thymine. And then sa purines, they have a similarity with each other. They are the adenine and then guanine. So, yun po. So, DNA as a genetic code. No? So, numerous studies have already proven that DNA is the genetic material or template for protein synthesis. This means that the DNA controls the cell structures and its functions. It exhibits 3 characteristics that will make you understand the protein synthesis easier. First, the DNA is the universal code for all organisms. This means that the genetic coding system is true for all living organisms, including viruses. Second, the genetic code is a triplet code. This means a triplet of nucleotides represents an amino acids to be included in the protein to be synthesized. An example is a triplet of adenine, guanine, and cytosine, which stands for AGC, is coding for serine. No? Malalaman natin mamaya yung genetic coding na yan. Then next, it was discovered that even the start code, which is AUG, and the stop codes, ito po, yung stop codes natin, ulitin ko, yung start code natin is yung AUG, and the stop codes, ito po yung UAA, UAG, at saka UGA, or para mas madali nating matandaan, uwa, uwag, uga, no? Uwag, uga. No, and are also in triplet of nucleotides. These codes are seen in the table of codons for amino acids. However, the table of codes using mRNA codes or codons because it is the mRNA that transcribes or reads the DNA codes. Third is the genetic code is a degenerate code. This implies that although a triplet code is specific for an amino acid, several triplet codes can code for a single amino acid. An example is GCU, no? Is coding specifically for alanine, But GCC, GCA, and GCG are also codes for alanine. Paano po nangyari yun? Meron tayong sinusunod na genetic codes. Ito na po yun, no? So, ano yung na-mention dito? Alanine. May GCU na sa alanine na GCU, and then na-mention yung GCA, GCC, at GCG. Under siya ng alanine. No? And then, sa DNA replication, this is a process of duplicating the entered genome prior to cell division. Extreme accuracy of DNA replication is needed in order to preserve the integrity of the genome for several successive generations. Reproduction at any level, either cell or tissues or organism, is impossible without DNA replication. So, meron tayong 4 steps ng DNA replication. So, they are initiation, priming, polymerization, and then termination. So, sa initiation, it is the unwinding of the 2 DNA strands. This process is catalyzed by DNA endonuclease and DNA helicases. So, ganyan yung nangyayari during initiation. Parang nag-unzip siya or nag-unwind. And then sa priming, ito yung attachment of primers, which are, attachment of primers, which are RNA nucleotide where DNA nucleotides can attach to. This is catalyzed by the enzyme primase. Then polymerization is the addition of new DNA nucleotides and are attached to the DNA templates, which were the original DNA that have separated in the initiation phase. This is catalyzed by DNA polymerases. Then yung termination is the stage where 2 new DNA molecules are formed. This process is catalyzed by DNA ligase and DNA topoisomerases. And then sa protein synthesis, it is the process by which the cells create new proteins for various purposes. The process involves the DNA, RNA, and the ribosome, or in some other textbooks, instead of ribosome ang ginagamit natin, protein, no, sa isang cell, no, or palitan natin yung ribosome into a protein. So we will learn how the DNA is used as a template for protein and how the genetic information. So, nakikita nyo dito is yung process ng central dogma ng DNA. From DNA, nag-downgrade into RNA, and then it will translate into a protein. Meron tayong 5 steps, no, sa proseso ng protein synthesis, which are the initiation, transcription, translation, elongation, and determination. All these processes are catalyzed by enzymes. Ano yung ibig sabihin ng catalyzed? Catalyze, it means that it is a cause, or sabihin natin an action or process to begin with, no? So ayan. So una, yung initiation or unzipping. A segment of DNA unzips. This is initiated by a cleave or a cut done by DNA endonuclease enzyme between a phosphate molecule and a deoxyribose sugar. This is followed by the unwinding of the DNA helix with helicase enzymes. So ayan na po. So, base sa nakikita ninyo, ayan yung process ng initiation processes, no? From helicases, unwind the parental double helix, single-strand binding protein stabilize the unwound parental DNA. The leading strand is synthesized continuously in the 5' hindi po 5' yan, 5' to 3' direction by DNA polymerase. The lagging strand is synthesized discontinuously. Primase synthesizes a short RNA primer which is extended by DNA polymerase to form an Okazaki fragment. Ano yung Okazaki fragment na yun? Okazaki fragments are short sequences of DNA nucleotides, approximately 150 to 200 base pairs long in eukaryotes, which are synthesized discontinuously and later linked, no, together by the enzyme DNA ligase to create the lagging strand during DNA replication. So, ito na po. And then number 5, after the RNA primer is replaced by DNA by another DNA polymerase not shown, the DNA ligase joins the Okazaki fragment to the growing strand. So, ayan po. Next po is in transcription. This is the synthesis of an mRNA or codon based on a DNA template through base pairing. RNA nucleotides bind with DNA nucleotides by complementary base pairing with now of the 2 DNA strands that are exposed after unzipping. Yung A, kasama niya yung U, and then yung C, kasama niya yung G. This is catalyzed by the RNA polymerase enzyme. No? So, this is followed by the movement of the newly synthesized mRNA from the nucleus out of the nuclear membrane and into the smaller subunit of the ribosome. Translation: when the mRNA attaches to the smaller subunit of the ribosomes, tRNA from the cytoplasm enters the larger subunit of the ribosome with a 3-letter code anticodon na UAC, no? Or UAC, ayan. No? So this tRNA carries the amino acid methionine or MET, no? Kung makikita natin dun sa references dun sa genetic code natin or sa mga codons, this signals the start of the protein synthesis. Note that the mRNA codons determine the type of amino acid brought into the ribosome by the tRNA. Then elongation is the repeated translation. This is accomplished with tRNA in the cytoplasm that continuously enters the larger subunit of the ribosome. The ribosome is large enough to accommodate 2 tRNA molecules at a time, one incoming and one outgoing. And then the amino acid on the incoming tRNA forms a peptide bond with the amino acid on the outgoing tRNA. The ribosome then moves laterally, allowing the outgoing tRNA to leave and making space for a new incoming tRNA. The polypeptide lengthens one amino acid at a time. This is still catalyzed by RNA polymerase. Then termination. It is also known as a translation process occurs at the stop codon on the mRNA. This is also catalyzed by RNA polymerase enzyme. The stop codon that can signal for termination are UA, UAG, or UGA. No? This, yun yung stop codon dito. No? Alam ko, nabanggit ko na to kanina. And then, this is translated by a tRNA but does not carry any amino acids. The completed polypeptide or protein dissociates from the ribosome and the ribosomal subunits separate from each other. And then ganyan yung itsura ng initiation, elongation, and termination process. So ayun lamang po. And then sunod na po tayo sa next topic natin na mutation. Mutation, no? It is a change in genes or chromosomes which causes a new trait to be inherited. Hugo de Vries, no? Pakicorrect nga kung tama ba ako dun sa pronunciation na Hugo de Vries, no? And then alamin natin kung ano yung tamang pronunciation ng pangalan niya, but anyway, babalikan natin yan later on. And then it is a change in genes or chromosome which causes yun nga, nabanggit ko na kanina, no? Medyo lutang lang. He coined the term mutation in 1886 after he noticed some remarkable changes in his experiment with the evening primrose, no? Wait lang po, alamin natin yung pronunciation ng pangalan niya. May mga nagsasabi na or Hugo de Vries, no? Tama ba ako? Hugo de Vries nga ba? Just leave a comment below kung tama ba yung pagkaka-pronounce ko. So anyway, ano yung evening primrose, no? Ito ba yung gabi? Gabi niya ba ito natuklasan? Hindi po, no? Yung evening primrose, ito po yung plant, no? Kadalasan nako-convert siya into an oil, no? Or langis. So, in terms of mutation again, if a mutation occurs on a body or somatic cell such as the skin cell, the mutation only affects the individual that carries it. Meron tayong dalawang klase ng mutation, no? This mutation is called somatic or acquired mutation. No? So, this kind of mutation is not inheritable and thus it does not carry the risk of affecting the future generation. Dun lang sa part ng katawan lang niya. Or parang sabihin natin, under lang siya ng somatic cell. No? Kung alam natin yung mga basics sa sexual at sexual reproduction, magigets natin to. And then, on the other hand, if the mutation occurs on a germ cell or sex cells, this is called germline mutation or heritable mutation. This type of mutation is inheritable and can be seen or passed in the future generation. No, so see, um, most mutations are harmful or negative mutation. No, that is, they reduce an organism's chance for survival or reproduction. For example, sickle cell anemia is a serious blood disease which is caused by mutation in a gene. No? So sickle cell anemia results in blood cells that are shaped like a sickle or half circle. People who have sickle cell anemia have difficulty obtaining enough oxygen to all the cells in the body. The sickle-shaped cells may clump and clog the blood vessels. So, ganun po yun. Some mutations are helpful or positive mutation and cause desirable traits in living things. When mutation occur in crop plants, the crops may become more useful to people. A gene mutation in potatoes has produced a new variety of potato called the Catadin potato. This potato is resistant to disease that attack other potatoes. Also, the new potato looks and tastes better than other types of potatoes. This type of mutation is the mechanism for evolution to occur. It may seem as if mutation produce only helpful or harmful traits. However, that is not always the case. Many mutations are neutral and do not produce any obvious changes. Still, other mutations are known to cause dead organisms. No? So in terms of point mutation, they are small changes often in the single nucleotides or single nucleotide base. The sickle cell disease occurs when the sixth amino acid, glutamic acid, is replaced by valine to change its structure and function. This is caused by the mutation of a single nucleotide T to A from a triplet CTC to triplet CAC codon, causing a change in the structure of the protein hemoglobin, which causes the erythrocyte carrying the red iron pigment to become crescent-shaped or sickled. So, next naman po is yung deletion. So, in deletion, it removes information from the gene or chromosomes. It could be as small as a single base or as large as the gene itself. Example nito is yung Chat or cat's cry syndrome, which is caused by a deletion of the end of the short or yung P-arm of chromosome number 5. This deleted portion of the chromosome causes gene to be missing. So ano yung Chat syndrome? Ayoko nang ipakita dito, no, yung mukha ng isang baby, no. So ma-consider ko yung characteristic ng Chat syndrome sa pamamagitan ng moon-shaped face, heart disease, mentally retarded, malformed larynx, but it has a normal lifespan, no. So the size of the deletion varies among affected individuals, and researchers suggest that the larger the area, no, area of deletions, the more genes are removed, resulting in more severe intellectual disability and developmental delay. In terms of insertions, it occurs, no, when extra DNA, genes, or extra base pairs are added into an existing gene. Example nito is yung Huntington's disease. The gene defect responsible for this disease is found in chromosome 4. It is a small sequence of DNA where several base pairs are repeated or inserted many times. Common symptoms ng Huntington's disease like hallucination, cognitive decline, mood swings, involuntary movements, speech difficulties, and behavioral and personality changes. So, ayun po yung yung symptoms ng ganitong klaseng disease. The normal gene has only 3 DNA bases of the nucleotide sequence CAG. For patients with Huntington's disease, the sequence abnormally repeats or inserts itself dozens of times. Sa frameshift mutation, it results from either insertion or deletion of 1 or 2 nucleotide bases that often create new stop codons and thus generate nonsense mutations. When this occurs, the reading frame is changed so that all the codons read after the mutation are incorrect, no, even though the bases themselves may be still present. An example of this is Tay-Sachs diseases. This genetic disorder is caused in most cases by a 4 base pairs insertion in exon 11 of the hexa gene found in chromosome 15 that causes a premature stop codon, leading to a profound deficiency of one of the subunits of hexosaminidase or yung hexa. No? So this protein helps break down a chemical found in nerve tissue called gangliosides. Ulitin ko po, gangliosides. The absence of this protein leads to gangliosides build up in cells, especially in the nerve cells in the brain. So, last one na rin, which is the mutagenic agents. Certain environmental factors can cause mutation to occur at a greater than normal rate. Such factors are called mutagenic agents. Such agents are radiation, chemical, and infectious diseases. Sa radiation, it's either X-ray, no, or UV radiation. Mas lalo na yung sa UV radiation, no. Sa chemicals, it's either smoking or vaping contains dozens of mutagenic chemicals, nitrate and nitrate preservatives, no? And then benzoyl peroxide, which is the common ingredient sa acne products. And then lastly, sa infectious agents, just like yung HPV or human papilloma virus or HPV, which is the sexually transmitted virus. And the last one is yung Helicobacter pylori, which is yung H. pylori. Mali yan, dapat naka-italicize yan. And then dapat ito hindi dapat capitalized, no? And then bacteria spreads through contaminated food. So it is not limited to the following na iyan lamang ang mutagenic agents. Marami pa po, no? So konti lang po yung naibigay nating example, but since dito na nagtatapos yung ating lesson, no? So kung meron kayong mga clarifications, questions, or concerns, leave a comment below. And then, so that's all for today. Kita-kits sa susunod na lesson. Paalam.